I'm Ian Hans Portman. I've been at Warwick now for 29 years, which is more than half my life, so either I love my job or I'm just too lazy to move. I'll let you be the judge of which. I'm a microscopy manager, a technical specialist, so I sort of act as a translator between the microscope and the researchers that are trying to use them. I didn't start off in microscopy, I actually started off in X-ray crystallography, then I did a stint in neurobiology and then neurophysiology, which is kind of the same thing as neurobiology but a bit more zoomed out. All these things have one thing in common, which is microscopes. So, about 15 years ago, when I found out the previous manager was leaving, I started making moves to get my foot in the door in the microscopy suite, which is where I've been ever since. I got my first microscope when I was 8. It was terrible and it cost £10, which was a lot to a kid in 1980. But you can still get them on eBay for £10, so it's still not good value for money. But it didn't put me off. My microscopes now have changed a bit. They're quite a lot bigger. This one takes up a whole room and it costs a little bit more. This was £900,000 and it's also full of lasers. Sorry, I grew up on 1970s prog rock and B movies, but I have to be responsible for laser safety, which I'm not so keen on, but it has to be done and I'd rather look after my own eyes than trust anyone else to do it. Sample prep's also a bit different. My 8-year-old method of squishing my thumb down on the cover slip as hard as possible doesn't really cut it. It takes a little bit more patience than that. This is me preparing plant specimens for antibody staining. It takes nearly a week to get from the plant to something on a slide you can actually look at. It won't be the only thing I'm doing that week, but it'll be something I have to check on every hour or so during the day for that whole week. Some of our reagents are also incredibly dangerous. To stain cells, we're using things like toxins from death cap mushrooms, things that block cells from making energy, and chemicals that glom themselves onto DNA and never let go again. You can't just lob those in the bin when you're done, so I have to keep records of that and arrange for proper disposals. And because we're known across the department for having some of the worst chemicals on campus, I also get called in when other labs have some sort of a chemical incident. But it isn't all fun and excitement. £1 million microscope is not a robust children's toy and they need a lot of care, especially if the students have been in unsupervised over the weekend, and there's a fair bit of tidying and cleaning to be done. When you're looking at tiny things, dust gets to be a real problem. It gets into everything. The tiniest little speck can cause us some problems. So the labs are going to be kept clean. Hunting dust bunnies is a weekly sport. We don't ask the cleaners to do the lab areas because we've got these fun chemicals, we've got lasers, we've got high voltage equipment, and did I mention pathogens as well? And more important than keeping the lab clean, the lenses have to be kept clean. So twice a month, sometimes more, I have to take each lens off the microscope, take it to another microscope, sit down and start cleaning it. I don't actually mind doing this. I stick on a bit of music and sit there with a pile of lens tissues and cotton wool buds and just pick away at the dirt on the lenses. If the lens is really bad, it can take over an hour, and usually someone's going to get an annoyed email from me after I've done something like that. But one of these bits of glass and steel costs us about £15,000 to £25,000, so it's definitely worth the effort. One of the major aspects of my job is the teaching, and I absolutely love it. There's a certain amount of self-selection because anyone coming to use the microscopes is going to be interested in what they're doing, so my trainees are usually very keen to learn how to use the equipment. Many of them are PhD students, so I get to see their careers develop and see their science develop over 3 or 4 years. And then sometimes I get to see them come back a few years later and start their own research groups, although they never seem very surprised that I'm still there. I love to learn new things. When a new project starts, I have to read up and try and understand what people are trying to achieve and what the biochemistry is in their cells and try and help them come up with the right approach. I can pretty much guarantee every week that I'm going to get to see something absolutely astonishing and beautiful, so I thought I'd share a few examples with you. Is endoplasmic reticulum in a plant cell. The ER is responsible for making and directing proteins around the cell. Everything we eat is made here. This is shot in real time. We're actually seeing proteins in blue being trafficked around a network of tubules inside the cell to distribute them, and in green we've got the chloroplast tightly connected to the ER. So you can see here that plants may not appear to move much, but inside they're a churning cauldron of biochemistry. Some pollen grains. I use pollen grains as a test sample for the microscope. There's a lot of detail on them, which makes them ideal for checking your resolution and checking how well everything's behaving. They also happen to fluoresce by themselves, which makes them much more convenient to work with in lab because I don't have to bother staining them, and they're quite easy to get hold of. They also happen to look stunning. One of the great things about a busy lab is the variety of things that we get to work with. We've had plant scientists, we get cancer researchers and material scientists as well. Sometimes I get things that people think might be a bit weird. This is cheese. I was helping someone who's doing a PhD in cheese making, and she's also a professional cheese maker. The blobs of fat and then the stuff surrounding it is protein. I've also looked at ice cream, glue, and even once, although I was never ever allowed to touch the sample myself, a piece of real Mars rock. Finally, rotifers, one of my favourite animals. I'm going to be using these for some school sessions on microhabitats and adaptation to the environment. I also wanted to show that not everything we look at is dead. Don't be fooled by the tiny size. These things have a complete nervous system, they have a brain, a gut, they're about 1,000 cells in all. This isn't strictly speaking research, but part of our remit is to educate and encourage people into science, so why not have a little bit of educational fun? Working with microscopes gives me an incredible sense of wonder almost every day, and I really hope that these tiny but complex critters might help pass on a little bit of that. Thank you for listening.